Welcome to The Explainer. Today, we're unlocking biotechnology, which is the science that quite literally rewrites the code of life. We're going to track the evolution of a field that completely transformed from a mere theoretical concept into a very real applied reality. It's a reality that actively shapes the food on our plates, the fuel in our machines, and the medicine inside our bodies.
Okay, let's dive into this by setting the stage with a bit of a brain teaser. What exactly connects genetically engineered glowing plants, a cloned sheep named Dolly, and a viral pacemaker that keeps a human heart beating? Now, on the surface, I know they sound like entirely unrelated concepts straight out of a sci-fi movie. But, as we'll see, they are all incredible milestones achieved through the exact same fundamental branch of human ingenuity.
To understand how those seemingly impossible feats are even doable, we really need to define our terms here. Biotechnology isn't a pure science in the traditional sense. It's an integrated effort. The term itself was coined over a century ago, way back in 1919, by a Hungarian engineer named Karl Erecki. And get this, he was originally just trying to find more efficient ways to raise pigs. He defined it as the application of technology using living organisms to create useful products. Essentially, it's about putting biology to work for us.
Now, because it's such a hands-on, applied field, biotechnology is truly multidisciplinary. It's almost like a giant puzzle. It draws heavily from your basic sciences. So, mapping out the blueprints using genetics and molecular biology, and figuring out how the systems tick via immunology and physiology. But here's the absolutely crucial part. It pairs all those raw foundational sciences with chemical and biochemical engineering. That specific combination is what allows biotechnologists to turn living cells into microscopic factories that can pump out life-saving vaccines, engineer super-resilient crops, or even treat industrial waste.
Now, what's really interesting about this slide is how the scientific community categorizes all these vast applications into a brilliant color-coded framework. For instance, red biotechnology focuses entirely on medical applications and human health. Green handles agricultural biotech, while white represents the industrial side of things. But it actually goes even deeper than that. We've got yellow for nutritional biotech, gray for environmental applications, gold for bioinformatics, and yes, even dark biotechnology, which deals with the rather grave realities of biological weapons. This color system really helps us organize the sheer, massive scope of everything biotech touches today.
So, those broad, color-coded fields, they are driven by highly specialized operational toolkits. You have traditional genetics, which deals with hybridization and selective breeding, but then you have the more modern pillars. Things like molecular biology handling recombinant DNA, cell culture driving stem cell technology, and bioinformatics managing the absolute mountains of data required for computer-aided drug design. Every single breakthrough we teased earlier relies on a very specific combination of these operational disciplines working together.
Let's transition over to the early breakthroughs, spanning from 1968 to 1980. To really grasp the magnitude of this field, we have to look at the timeline of discovery. Starting in 1968, scientists conceptualized and then soon after discovered restriction endonucleases. Now that's a mouthful, but essentially they're molecular scissors used to cut DNA. Within just a few short years, they used these tiny scissors to produce the first recombinant molecules and develop hybrid plasmids. This rapid, frankly breathtaking acceleration of genetic tools culminated in a massive real-world milestone in 1980 when Eli Lilly produced commercial human insulin using genetically engineered bacteria. For the very first time in history, we were literally using living cells as factories to produce a vital human hormone.
Up next is the genomic revolution, covering 1986 to 2003.
And this brilliantly illustrates how, once basic genetic engineering was proven commercially viable, the field just absolutely exploded. I mean, in 1986, the discovery of the polymerase chain reaction, or PCR, basically gave scientists a biological photocopier, allowing them to rapidly amplify DNA. That directly set the stage for launching the monumental Human Genome Project in 1990. Meanwhile, biotech was hitting the consumer world. 1994 brought us the FDA-approved Flavor's Sauver tomato, and 1997 gave us Dolly the sheep, which proved once and for all that adult cell cloning was entirely possible. By 2003, we had successfully mapped the locations and sequences of genes across all 46 human chromosomes.
Now let's move to and see how this builds into the CRISPR and beyond era, from 2008 to 2023.
With the human genome mapped, we expanded straight into the realm of science fiction. Just look at what becomes possible when technology and biology fully synchronize. By 2009, modified heart genes were used to create a viral pacemaker. By 2012, bionic limbs were being successfully controlled directly by the human nervous system. Then we hit the true game changers. Starting around 2019, CRISPR technology was deployed to edit human genes in an attempt to treat cancer where standard treatments had completely failed. And researchers began introducing prime editing, which is a tool vastly superior and more precise than early CRISPR methods.
I want to pause specifically on the year 2020, because it truly ushered in an entirely new reality for us. In that single year alone, scientists reported using CRISPR-Cas9 gene editing inside a living human's body for the very first time. They developed artificial chloroplasts, and they even engineered plants to glow brilliantly using bioluminescent mushroom genes. What was considered absolutely impossible magic just a century ago is now a documented peer-reviewed reality. We are actively writing and rewriting the biological software of life on Earth.
So, what does this all mean? The crucial point is this, and I really want to leave you with a final thought to chew on. Carl Ehricke first defined biotechnology in 1919. In exactly 1 century, we went from merely conceptualizing this fusion of biology and technology to literally editing DNA inside a living human body and building thought-controlled bionic limbs. If that is what we managed to accomplish in the first 100 years, what on earth will the next century of biotechnology look like? Thank you so much for joining me for this explainer. Keep questioning, keep learning, and I'll see you next time.